Okay. So let's look at an illustration of what we have been talking about. So this is a whole muscle, the whole muscle. Of course, skeletal muscle. Why are we saying skeletal muscle? Because we can see that there is a tendon on both ends. This side there is a tendon, and the other side we are seeing a tendon. And that tells us it is that this muscle is attached to a bone. Good. A skeletal muscle, or all muscles generally, are composed of muscle fibers, which are called muscle fibers or muscle cells. Now, this is how a muscle fiber looks like. It is a cell, but which is looking funny because it is long and the names of their parts are different. You can see it has many nuclei. This is one nucleus, this is another one, this is another one, and there is another one here. So it is multinucleated. Then it contains, of course, the sarcoplasm. You know what it means, the cytoplasm. Then the sarcolemma is its cell membrane. Then, of course, it has these striations, which we are tracing from the myofibrils. Those striations, please, they are very important. Now, of course, the striations, you can see some of them are white, some of them are dark. Now, let's pick one muscle, myofibril, and look at it. One myofibril. Now, each myofibril, this one is just a very small cut portion of a myofibril. Just to illustrate, we have just cut a small portion of a myofibril. So, when you look at a myofibril, each of them consists of a light zone, a light band, not a light zone. This is the light band, which appears light. So, from this point up to this point, we call it the light band. And then from this point up to this point, this one here is the dark band because it appears dark. Then after that, there is another light band. What do you expect after this light band? We expect a dark band. After the dark band, we expect a light band. After the light band, dark band, etc. like that. Now, in the middle of the dark band, there is this region here, from here up to here. Does it look the same way? Does it look dark the same way this portion and this portion are? You guessed it right. No. This part and this part are looking darker than this middle part. So this middle part, we are calling it the H zone. H zone is lighter than the other regions of the dark band. And of course, the H zone is also found in the dark band, but it is lighter. Good. Then, in the middle of the H zone, there is a dark line. There is a thick dark line which runs in the middle there. We call it the M line. We call it the M line. Then, in the dark band, in the middle of the dark band, the light band, sorry. In the middle of the light band, we see a line, again a thick line, running in the middle of the light band. That line is called the Z line. So even this side, the middle of this light band, we are seeing another Z line. And we are saying the region between this Z line and this Z line is what we call the sarcomere. The sarcomere. So what is the sarcomere? The region between two Z lines. So we say that this one, of course, is the dark band. If we started from here, we would have another dark band here. But we know that in the middle of the dark band, there is another zone which is lighter, which we call the H zone. Then in the middle of the H zone, there is a line, a thick line, which we call the M line. Okay. Then if this is our light band, in the middle of this light band, here in the middle there of this light band, we have the Z line. Remember this one? What was this one? This one was our M line. And the whole of this was our H zone. So if we wanted to get a sarcomere here, where do we start from? Sarcomere, we start from this other Z line and we move up to this next Z line. This gives us a sarcomere. So that is a myofibril, a portion of the myofibril. Now, what are the myofibrils? What are these dark zones here and the light zones made up of? What are they really made up of? There are only two proteins which we said are in a myofibril. They are the thin actin filaments and the thick myosin filaments. So, when you are representing them, we always draw a central line. Draw a line there. Okay? That thick line in the middle represents the M line. Represents the M line. Then, from the M line, we have the thick myosin filaments coming from it on both sides. So you draw, from both sides you draw the thick myosin. You draw them as thick lines. Then, on the other side, we have the thick line which is representing the H line. This, rather, not the H, the Z line. Even on this side, there is a Z line. You draw it there. Then again, you draw thin lines radiating on both sides of this. Okay, you draw that radiating on both sides. However, you don't make them all to go up to the middle. You don't make the thin lines to go up to the middle. You can see they are stopping here. The thin lines are stopping here, and then there is some space between this and that. This space here should remain. So let's try to analyze. Let's try to analyze. Let me first remove this, and we try to analyze the thing very well. Let me first remove this. Okay. Now, remember what we said? We said that the light zone, the light band, not the light zone. The light band is composed only of the thin actin filaments. Let's look this side here. So where is the light band starting from? It is starting from here. I'm trying to compare it to the upper one, this one. So you can see that it is only these thin actin filaments, the thin ones, which are found in this dark band. Rather, the light band, in the light band. And there is a thick line in the middle of the light band, which we call the Z line. Then, the dark band contains the two regions. There is the middle one. There is this middle region starting from here